So now we're getting on to some proper science for this session. For those of us who are clinicians, you may have to have a cup of coffee and a little bit of focus. And we're having a run through various aspects of translation and medicine. I'm just going to check with Dirk one thing. Dirk, is everyone here?
Ja, stationair.
Open doen?
Ja.
So, there's been a lot of interest, obviously, in NIPT testing. I think first reported here in Leuven, I think, diagnosing ovarian tumours on NIPT, because it obviously does interfere with those results. So, first up, we have Joris Vermeesch. Your name is, for an Englishman, is very hard, Vermeesch. But forgive me if I've got it wrong, sorry. Who's going to talk about NIPT testing to detect ovarian cancer, to detect cancer in pregnancy. Joris, over to you.
Okay. Good afternoon, everyone. I always, when I'm invited to speak, I start by saying, thank you, what a beautiful city this is. I've never been here before. But I guess, because I'm from Leuven, I cannot say that today. But anyway, I'm very grateful to be invited here, because while we go to speak all over the world, it's very rare that we have the opportunity to talk at home. This is obviously an international meeting, so most of you don't know, but it's also a pleasure for some of the people here who might be local to be able to show our work. So today I will talk about NIPT in pregnancy, and I think this is an audience of gynecologists, so I don't need to explain what non-invasive prenatal diagnosis is, but I will do a little bit of that. So maybe first everyone knows that the fetus sheds some DNA in the bloodstream. It's not the fetus, it's of course the placenta. There are the placental cells which go in apoptosis, which actually release the DNA in the maternal bloodstream. And then if the fetus is normal, of course, we don't see any patterns, but if there's a fetal aneuploidy, we can actually detect these small differences of fetal DNA, the fetal DNA in the maternal plasma, and we can actually deduce whether or not the fetal is carrier of a trisomy 21, 13, or 18. And because fetal aneuploidy screening is a major, has been a major aim of prenatal diagnosis, it's clear that having an accurate test to do this non-invasively would benefit both the families and probably healthcare in general. And so, since the discovery of free-floating DNA in maternal blood, which was made by Denis Lowe, an observation made by Denis Lowe while he was still a postdoc at Oxford in 1997, I think, since then, the idea has grown that we could detect fetal aneuploidies, but it was until 4 or 5 years ago, in 2011, time flies, 6 years ago, in 2011, that it became actually possible to do that. And it became possible thanks to new instruments, which we had in genetics, which were called the so-called next-generation sequencing, I would call them today, massive parallel sequencing. And basically what we do is just sequence millions of fragments which are present in the maternal bloodstream, and then do some calculations and statistics on it. Actually, if you look worldwide, there's basically two big methods which are being implemented. One is what we would call targeted resequencing of DNA fragments. In this targeted resequencing, we have two major approaches. One is targeted resequencing of single nucleotide polymorphisms. These are variants which each of you is carrying. We're all a little bit, we all carry 46 chromosomes, but each of these chromosomes contains small variations, and catching those variations allows us to discriminate which alleles are present from the mother and to deduce which alleles are present from the father. And paternal alleles in a maternal DNA background are obviously from the fetus. This is basically the approach. So you amplify around SNPs and then start sequencing those fragments and doing some counting statistics. And this is the method which is used by Natera. We have another approach, Ariosa, which is basically locally amplifying fragments on mainly the chromosomes of interest, 13, 18, and 21. Actually, this is also usually on the chromosomes of interest, a number of controls. The fragments are amplified, and then there's another probe which detects those fragments and is used to count, again, the number of fragments of each of these chromosomes present. And then there's counting statistics to deduce whether there's an overrepresentation or not of that particular chromosome. But what the other groups are doing, and this is mainly the Verinata approach, or now Illumina, became Roche, actually. So the Verinata approach was to just randomly sequence, no target enrichment, just take the DNA. Actually, as a scientist, and I'm a scientist and a technologist, it was actually the easiest experiment I've ever done. You just extract DNA, throw it in a sequencer, randomly get fragments, map those back to the reference human genome, which we have, and then count. And that's basically, we align the reads, and again, we use counting statistics to see whether there's an overrepresentation for a trisomy or not, as compared to a series of references. So I would probably not be here if it wasn't that maybe I first briefly say, we adjusted that protocol a little bit to improve the protocol, and this has now been shown by a series of publications, but also some colleagues who have shown some why these improvements to the method improve overall NIPT diagnosis, and I will briefly comment on it. But this is actually currently the analysis pipeline we're applying here in Leuven and for the majority of Belgium. So there's counseling, or more and more as gynecologists, there's some leaflet and information about the pros and the cons of this technology. We sample the blood in so-called Streck tubes, which stabilize the maternal blood cells, because the maternal white blood cells are still there, and if you don't stabilize those, they start being degraded, releasing DNA in the bloodstream, and you cannot detect the fertile fragments anymore. So we do the plasma isolation, extract the DNA, make a library, then this is thrown in these massive parallel sequencers, and we get our results. Just very briefly, how our results look like is like this. Rather than just presenting a risk for trisomy 13, 18, and 21, we've done something else. And this is the novelty. Rather than showing the overall statistics of the chromosome, we actually separated the chromosome in bins of 50 kb, just map the reads in these 50 kb bins, and then plot the probability not of the overall chromosome, but of each of the bins. And of course, if you bring this all together, the Z-score, and this is what some of you may know, the Z-score here in this particular example of a trisomy sample is actually above 3. Here it's 7.3. So we know that this is a trisomy in the fetus. And I will show you later why this plotting becomes important, because you will see that we cannot only deduce whether a fetus has a trisomy, but we can actually deduce whether it's a maternal copy number variant or whether there's a segmental fragment. Sometimes we don't have whole chromosomal deletions and duplications, but segmental. And this allows us to do that. We also have some other statistical measures which allow us to measure quality, but I think for this audience, this might be bringing us too far. So this was, that this approach was actually of value was also picked up by Agilent, and they licensed our approach and are now selling this worldwide. So anyone interested can actually use that one-site approach. So just to give you a slight view of what we've been doing thus far, we started end of 2013 implementing this clinically, and we have now done about 20,000 samples, and this is the outcome. You see we have quite a pickup rate. We have different reasons of referral. In Belgium, people have to pay for the majority, but a number of insurance companies or insurance systems are reimbursing those samples. I should also say, I think we're probably the cheapest in the world. It's €290 for a test. And you can see the results. Overall, we have an excellent result. We had no false negatives for trisomy 21 and 13. We have one false negative in 20,000 for trisomy 18, actually. In terms of false positives, we had 6 which were not confirmed, but for the large majority, we think that these are due to biological mosaicisms, and I will come back on that later. So this is nice, but because we screen the whole genome and we don't use this standardized approach of the companies, we actually also plot all probabilities for other aneuploidies elsewhere in the genome. And what you see here are all the trisomies we've been picking up and also some monosomies, which fetal aneuploidies which exist and just occur during pregnancy. And some of those are quite common. Trisomy 16, for example, is the most common reason for miscarriage. So maybe there's not so much benefit, you would say, because these fetuses would miscarry anyhow. But look here, trisomy 15. Has anyone seen ever a trisomy 15 during? Probably not. But has anyone seen a Prader-Willi syndrome patient? Do you know what Prader-Willi is? I think so. Eh? These are people who have, they're eating and they get very obese and they have an eating disorder such that they basically eat themselves to death. That's due to uniparental disomy of chromosome 15. And what is uniparental disomy? That's if 2 chromosomes come from one parent. And in this, for Prader-Willi, it's from the mother. So if you have your both chromosomes from the mother and none from the father, that causes UPD. Well, that UPD is usually, first you have a trisomy, you have a loss of a chromosome, and the fetus, only the cells with 2 chromosomes 15 survive, and you end up with Prader-Willi. And we've shown in some follow-up cases that there was mosaic for UPD 15 in the patient. So we avoided a very severe disorder. I would say that's more severe than Down syndrome. The same is true, I think this also, I believe this analysis improves pregnancy management because a number of these trisomies are associated with intrauterine growth retardation. For example, trisomy 7, which is actually quite common. Actually, UPD7 can cause Silver-Russell syndrome, again, a very rare developmental disorder. So I think this improves pregnancy management. Not surprisingly, because we plot these segments along the length of the chromosomes, we can pick up segmental deletions or duplications. And here's a nice example where actually the Z-score, if you would have done the test elsewhere in the world, you would have had a Z-score of 7 and have thought this is a trisomy 18. But you can immediately see that not the whole chromosome 18 here is amplified, but just this little tip here. And what is this? Well, this is a special chromosomal abnormality, and it's an isochromosome 18. This is actually the karyotype following amniotic sampling. So first we did the array on the amniotic fluid DNA, and then the karyotype, and you see that this is an isochromosome 18. An isochromosome 18, of course, it's almost as lethal as trisomy 18, but nevertheless, this is different. And we've seen a number of imbalances which we've followed up and which were shown upon follow-up that actually we had some severe chromosomal abnormalities. For example, I forgot to mention, this is not in here, but we had, for the full trisomies, we had one full trisomy 2, which was then invasively followed up, and the fetus had actually a marker chromosome of chromosome 2, which involved many genes and would lead to a very severe cytogenetic disorder. And here we show a number of other imbalances. So I think this is very nice. In addition, we can actually detect maternal CNVs, which might be relevant for the mother with deletions or duplications. Here's an example of the mother carrying an unbalanced translocation chromosome 3 and X. And actually, the mother is perfectly normal, but of course, if she inherits this to the fetus would actually be severely affected if it's a male fetus with a partial X chromosome. In this case, we could assure her that the fetus was normal female, but it's important for future pregnancies. And here's an example of a small imbalance, which actually involved only a single gene, RUNX1, but that can cause a bleeding disorder. And again, we reported this back to the families and also the gynecologist, because during delivery, knowing that this was the case was important. Of course, you may wonder, should we transfer all that information, and as a gynecologist, or should we do that? So we have to balance reporting back to the mother, increasing anxiety to family, and maybe to do too much follow-up. So we're careful in doing that, but I think in these 20,000 cases over 3 years' time, we have about 10 families where we have follow-up. Over these 3 years' time, we had only 10 mothers where we reported some segmental imbalances to which we think are clinically relevant. All right, but you're thinking this is a session on cancer in pregnancy, so I have to hurry up. We have a high success rate. 1% of the cases, we have to do repeat sampling in 2%, and in 1% of the cases, the repeat sampling is also bad. But usually, no, the repeat sampling usually is good, and that's what I'm showing here. The repeat standard deviation, the first sample is bad. We do a repeat sample, and everything is normal. But you see some cases where the repeat sampling is, again, bad quality. And when we saw this the first time in the beginning of 2014, I show you the example. This was actually the plot, the genome-wide plot of the case. The second sample was this one. And if you look close, you can immediately see that actually the pattern of imbalances we were detecting was identical. And I thought, well, this is impossible by chance. There's some biological reasons for that. And at that time, we didn't know yet what was going on. So we checked first the maternal blood was perfectly normal. We thought maybe it's the fetus, but we didn't really believe it. We got an invasive test, and she was perfectly normal. So then we said, okay, this must be something else, probably a tumor. We discussed this with the oncology unit, and lo and behold, so we had these investigations. The ultrasound for breast was negative, but then the whole body MRI showed some of these nodules here, which was indicative of some lymphoma. Then I got a biopsy. We obtained biopsies from these nodules, and the surprise was that the RACGH was perfectly normal. I thought we will find the imbalances we've seen with the NIPT, with this non-invasive procedure, and the fragments we're detecting are probably from the tumor, but everything was normal. I couldn't understand until we went back to the pathology, or we got the results from the pathology unit, and it turned out that this was actually a non-Hodgkin's lymphoma, and non-Hodgkin's lymphomas are characterized by these basically Reed-Steinberg cells, which are attacked by the immune system. And there's only a small percentage, 1% to 2% of the cells are actually, let's say, cancerous cells. And it are exactly those fragments from those cells, from these Reed-Steinberg cells, that are actually going in apoptosis. And I show you here the result. We could then prove that exactly, and only the Reed-Steinberg cells by FISH, that they correlate with the imbalances we could see in the NIPT. And that was our first proof and indication that this genome-wide profiling for during pregnancy could actually also detect a cancer. This woman got actually treatment during pregnancy, and this is the sample after the 3 weeks, after 2 weeks, I think, of treatment. And you can see this shot is later, after 2 months of treatment, I think. But even after 2 weeks, we could know that the treatment was working and that the Hodgkin's lymphoma was gone. This leads, of course, to the question, can we use this test just to detect Hodgkin's lymphoma and to study them? And here's just a proof of concept study where we actually then got samples from 9 other people who had Hodgkin's lymphoma, non-pregnant, obviously, and we could, in 8 of the 10, actually, we could detect patterns which were consistent with the Hodgkin's lymphoma, and also we can follow them up after treatment. Here's another example where we had, again, an abnormal NIPT profile, and here you see we had 4 blocks, large segments of the chromosomes, which were actually either duplicated or deleted. This was actually, we referred this woman to the MRI. and she was shown to have this lymphadenopathies. It's a follicular lymphoma, and when we got biopsy, you can see here the area CGH pattern and the karyotype, and you can see that this chromosome 6, 7, 12, and 13 are coming back. This 11, which we detected in this karyotype, is not present in the NIPT, and that's, of course, because some of these lymph nodules are metastatic, and they have different chromosomal imbalances as compared to what we detect in blood, which is the mean of all these nodules, of course. In this case, nothing was done. This is not. This is a latent lymphoma. We didn't do a treatment. Since then, this is actually the outcome. In these 20,000 samples, we've now had 7 cases, 7 patients which were referred to the MRI unit, and for 6 of them, a diagnosis for a tumor was made. So I think what this shows, because we had some critics, especially in America, saying, okay, you will send, now we have to send everyone with an abnormal NIPT result to the oncology unit, and you create a lot of anxiety. And I got even phone calls from abroad of people who say, I have a bad NIPT result, should I send them to the oncology unit? The answer is, I don't know the answer, but in our test, and the way we use the interpretation of the test, I think it's very specific. We have 1 false positive. This woman, we're following her up, but we have absolutely no indication what could be the question. So this, of course, these findings lead to a number of other questions. First of all, we're now testing whether we can use this method, and I think the next speaker will present some data where he uses this to study tumors, but we're also looking at the sensitivity of the test in pregnancy and whether we can use it to do response. And so for this study, we're collaborating with Frederik Amand and the whole oncology unit here in Leuven to get samples of pregnant women who were, when they got pregnant, actually have cancers. And so we're following those up. And the aim of the study is to have 40 breast, 20 hematological cancers, 20 cervical, and 20 ovarian cancers. And this is already some intermediate results I can show you. So thus far, these are the number of samples that have been collected within this consortium. It's also a larger consortium. I forgot to mention, it's not only Leuven, but I think it's this broader consortium on cancers in pregnancy, which is actually delivering the cases. And here you see that actually the success rate or the sensitivity is not that high. Every number here is another case, and only when it's red, we had indications that there is a cancer in those pregnancies. And you see that they're rather low. Not surprisingly, the larger the tumor size and the higher the grade of the tumor, the more likely that we can detect them. And we're following that up. For one case, so some of these women have surgery, but we've been following them up after surgery, and in one we could predict relapse actually. So it's nice that this methodology can then be used to monitor cancers even after treatment or after surgery and see whether or not they come back. Then I have only one more. The question we have now, well, if this is a good test for pre-symptomatic cancer screening in pregnant women, why not do it on everyone in this room? And the answer is probably we should. So we're actually testing that. We're testing that in a population where we think that the incidence of cancer is the highest, women or men and women older than 65. And actually, we're doing this particular study. It's a little bit too early to say, but we are picking up cancers. So I think there's great promise for society there. So in conclusion, I think I showed you that this genome-wide profiling during pregnancy is fabulous for fetal aneuploidy detection. Not only that, I think it allows for broader pregnancy management if we go beyond the 13, 18, and 21 trisomies. I also showed you, I hope, that we can detect pre-symptomatic cancers in those pregnant women. Actually, the estimated incidence of this, or the published incidence of cancers during pregnancy is 1 in 2,000 pregnant women. Our findings are exactly that number. So we have in 20,000 samples, we have 7 cases, 6 proven. It's a little bit lower. It's 1 in 3,000. But these are, of course, the unknowns on top of the knowns. And then finally, I think this approach will be has great promise for for just public health. Thank you very much. No, just. And I need to, before I forget, I need, of course, to thank everyone, especially in the gynecology unit in general and the gynecological oncology unit specifically. Yeah, thank you. Yeah.
Thank you very much. Any questions from the audience? I mean, do you think this is going to have a high enough sensitivity at the end of the day? I mean, if you look at Stephen Tong's paper, he had a sort of a retrospective case control study quite recently, and I would have thought his sensitivity was relatively disappointing in a sense for ovarian cancer. So what's your views on that? Because you talk about screening the general population.
Yes.
You've really got to increase that massively to really work.
But there's two answers to that. First of all, we have some ideas, I think, in the community, the technical community, some ideas to improve that sensitivity. If you look with what I showed you here, we're missing a lot, and you're absolutely right. We're missing a lot, but of course, if you look at it otherwise, we don't have many false positives, so your specificity is high. At least you catch those. But I do agree there's a lot to do around sensitivity,
in fairness,
so we're
people
not
want
there
a test
yet.
that detects cancer, not tell them they haven't got it. I mean, it's a tricky one, isn't it?
Yes.
I mean, because the two are obviously balanced. And I mean, looking at the NIPT platform in itself, the whole genome thing doesn't necessarily, you have to actually get down to subtypes of chromosomal abnormality almost before you start getting higher detection rates. So the standard platform doesn't seem to pick up quite as many as one would hope. Is that a fair comment?
Sorry,
Does the standard NIPT testing pick up the cancers you'd expect
so
them to
the most, I have to be careful, because for us this is the standard test, of course, but most commercial platforms, as far as I know them and that they're being used at the moment, do not.
Yeah, that's what okay. I think it's important for people to think they're not picking up cancer by running it through their NIPT testing. It doesn't work that way, does it?
That's true. What did happen is, however, there's a nice study of Diane Bianchi, who actually used the Verinata data for the first 20,000 from the study, and she went back to look at the data where there were some false positives. So they had a number of false positives. She looked at the overall genome-wide view, and then could show that a number of those, in 8 of the 30 false positives they had, that they were actually proven cancers, because then she went back to the health records and could show that later on these women were actually diagnosed with the cancer. So what that tells us, and I think that's important to remind, that people using those other tests in case of false positives, it is worthwhile to open up that box and look at it. And I think, I'm not sure whether Illumina people are in the audience, but I think they've been listening to me. So.
Okay, well, thank you very much. I think it's going to become a big issue because it's an emerging field. Thank you so much for your talk.
Thank you.
Okay, our next speaker is Liesbeth. I always know it's short A. Is it Lagae? Lagae. Yeah, thank you. There we are. Liesbeth is the R&D director and is co-founder of life science technology activities IMEC. IMEC, for those of you who don't know it, in Belgium is an institute of essentially nanoelectronics. I think I visited there with Dirk, looking at their chip technology. So they're really innovative and interesting group. So really interested to hear what Liesbeth has to say, and she's going to be talking to us about circulating tumor cells. So these are, different themes really, but translational approaches to looking at ovarian cancer detection. And very nice scenery as well, by the look of it. How are we doing? Perfect. Slightly shaky, but I think we're on. Dirk, did I hear right that there is ethics for that in IOTA 5, not just trans IOTA?
have approval for iota 7,
Right.
so, and that's trans iota. But that was in Leuven only because logistically it was not possible to expand it to different centers because you need to have the Streck tubes, you need to process the logistically quite
It's quite
difficult.
demanding, aren't they? The SOP is quite demanding for collection of these samples,
Yes.
isn't it? So it's
Yes.
not so easy for everybody to
So
contribute.
biomarker research is much easier because then you simply have to take a blood tube, so now we collect blood, serum, and plasma, and this should be processed very quickly, and it should be according to a certain protocol because even in relation how you handle the samples may affect the circulating DNA.
Are there any questions for, if any of the speakers are still sitting here while we're waiting for this?
Maybe you can come back on stage.
You just thought you were safe, didn't you? You know,
Yes, so
sitting there quietly.
these seats are much more comfortable than there. It's Yes, we can have the discussion in the meantime, so please have a seat. And maybe you can get another microphone. So these boxes, actually these are microphones, so don't be afraid of them. And just one quick reminder, during the coffee break, your last chance to visit the booths and to hand in your paper. And there will be the prize draw at the reception at 6 o'clock, so the winner will get another beautiful prize. Yes. So, any questions from the floor to our eminent speakers? Professor Valentin?
Do any of these things, do any of these approaches, do you think, are likely to give insights on treatment? You know, and response to treatment, because you know when we look at, we've always been tending to look at biomarkers, even HE4, and say, well, if your HE4 is X prior to treatment, what is your prognosis going to be? So if you look at your circulating DNA, are there aspects of that profile that might allow you to predict treatment response or relapse or likelihood to have resistance to chemotherapy, things like that? Are these things you think you may get insights on?
Yeah, I think you rather do this with mutations for specific targeted therapies, but maybe again ovarian cancer can be an exception because you have a lot more chromosomal instability in BRCA deficient tumors, which are treatable with PARP inhibitors. So maybe if you can very reliably quantify the chromosomal instability in the plasma DNA, you can also predict response to PARP inhibitors. So that's maybe one of the ideas that come immediately to my mind. But that's when you focus on chromosomal instability. When you focus on mutations, EGFR mutations and lung tumors, it's a very obvious example where they are doing it in the diagnostics already.
Because the holy grail is diagnostics and individualized care, isn't it? To try and put the whole thing together, not just looking at purely getting the diagnosis.
Yes, I agree, yeah. But with the chromosomal instability, you're limited, of course, because chromosomal instability by itself is not a predictive biomarker for most therapies. So, yeah.
Yeah. How are we doing on the technical side? Are we in business? It's a bit like being seasick if you're watching it. You might just... Question from Lil Valentin, yes.
I would just like to ask Joris Vermeesch, because I didn't quite get it. The method that you use, is it unique in Europe? No one else does it?
It's not. It was unique in the sense that the difference, it's random sequencing. It's actually similar or the same method as presented by Dieter. In that the difference with what most companies have done, and I have to say most because more and more units in Australia and also in Europe are taking up this approach. The difference was that rather than taking a whole chromosome and look at the Z-score of a whole chromosome, we divided it up in small pieces and looked at intermediate values. And that allowed us to get these chromosome-wide plots and see much more, and that provided much more information. I think the other difference is, of course, that we had the luxury, being in an academic center, to have the study expand to all chromosomes, whereas the aim of the companies was 13, 18, 21, and they didn't want to be bothered with all the rest. Whereas we had a different approach and viewpoint that this might be valuable information. And I think that's now percolating very slowly. But I think it will percolate further that's probably a better approach. But of course, society has to decide, gynecologists and maybe even patients have to agree that this approach is clinically valid and utile.
Thanks. Thank you very much. Guys, thank you. You're off the hook now. Honestly, we're not going to get you back on stage again, so you can relax. Liesbeth, I'm very sorry about the technical problems. It's never nice when you're speaking for that to happen, but please, over to you.
Yeah, so thank you, gentlemen, for filling up the time. I'm going to talk about something very different. I'm going to talk about smart silicon chips, and I'm going to talk about how they potentially can be used to sort circulating tumor cells. So it will be very different from what you're used to. So I will need to explain what smart silicon chips are as well. But before doing that, let me give you a view on where smart silicon chips are heading to. So this is probably a picture that looks familiar. It's a clinical lab, or supposed to be a clinical lab. And that's how people analyze samples today. So by 2020, it is expected that there will be 20 billion devices connected to this world. So this evolution is called the Internet of Things, and you probably have heard about that. But what does it mean for health? So will we also have connected devices that are analyzing your blood through the world? It will probably take a little bit longer than the Internet of Things, but we are convinced that this evolution will happen. So Internet of Health, things will come, and there's actually 3 parts, or 3 reasons why Internet of Things will become important. So let's take the view of the patient. So the patient wants to be more empowered, wants to be more quantified, and wants to be more healthy. Let's take a look at pharma. Pharma is actually interested in getting their R&D much more cost efficient for more precise medicine. And let's take a look at the therapy and at production of therapies. Well, also they are very much interested in getting very high quality of administration. And in all these fields, there is a push for Internet of Health things to come. So patients, they want precise decision taking, and they want quality data, and they want to see their data today. So the patients themselves are pushing for this to happen. Regulatory is pushing because they want to have much more quality tests and more data sharing between clinical trials. And then also in another field, in ATMPs, there's actually at this moment a strong push for starting to use Internet of Health things because they really have to think about scalable, distributed production of cells. And that requires quality testing and in a distributed fashion. So Internet of Things can come to the rescue. But how or what do you need for such evolution to happen? You basically need chip technology, and so that's where my world comes in. So my world is IMEC. IMEC is a research institute which is a world leading in nanoelectronics, and it's just across the street, actually, 1 kilometer from here. So we build, together with industry, very relevant solutions in electronics, and we diversified to healthcare about 5 years ago. So we are a research institute with a government subsidy, but with a lot of contract research for companies. And these are a number of the companies we work with, and you'll notice some, let's say, companies that you know as well, such as Intel and Samsung, are part of our network. So what is semiconductor industry about? Well, they are going all about miniaturization because this is just the most cost-efficient way of building systems. So miniaturization of chips, and you see an example here, was predicted by a guy called Moore 50 years ago, and for 50 years we've been keeping up with his predictions So he is our hero. But what does miniaturization mean? Well, it basically means that you can build a computer, which in the 1950s filled a whole room, into a Samsung smartphone, and I should update my picture with the newest model. So it's 10 to the 9th more powerful, 100 times smaller, and 10 to the 4th times cheaper than this original computer, which would never have made it into your pocket. So again, can we do the same for our healthcare test that we use every day? So we believe we can use those chips, and we actually have extended our silicon chip platform with a number of layers which are specifically intended to build the new generation of life science sensors. So on top of the conventional customized electronics, we have integrated a few additional layers. One deals with sensors, so with electrodes, but also with MEMS-based heaters and other structures. One deals with integrated optics, so with lenses, with filters, with a number of light excitation structures, and so on. And one deals with microfluidics for sample manipulation. So this smart silicon platform is now being used already in a number of commercial systems, where IMEC was part of the R&D. So our first example is a biophotonic sensor that is currently commercialized by a company, Genelite, for doing a 128 immunoassay tests from a drop of blood in 15 minutes. And the silicon chip is that small, tiny thing inside, which costs only 5 euro cents. Their instrument is still pretty big. They could simplify it further if they would put more functions on the silicon chip. A second one is, I think, one that got a lot of attention. It's a sequencing company, Pacific Biosciences, that also had an instrument that filled the whole room, and that, thanks to a development together with IMEC, could make their instrument 7 times faster, 3 times smaller, and at 2 times lower cost. And basically, the footprint has been reduced from half of a room to, let's say, a fridge. Again, thanks to smart silicon chip technology. So that is really the power of smart silicon chip technology, and is what I would like to try to convince you of. However, we need to tackle a next challenge, and for our next challenge, we chose cancer cells. So if you look at today's practice, this is your business. You know that better than I do. So let's summarize that there is a trend towards liquid biopsy, because that is just a very non-invasive way of testing with very precise information that can be obtained from that. And there's a trend towards doing genetic analysis of the cell-free DNA. If we look at the cells which are also present, the cancer cells, the circulating cancer cells, which are also present in those samples, there's much less work on that, and there's much less work on actually looking at the molecular information that is contained in those cells. Now, the reason, or one of the reasons that people prefer or first have explored the cell-free DNA as opposed to the circulating tumor cells is actually that DNA is much more stable, and that it's much easier to standardize the workflow, and that's, of course, a very good reason. However, if you would look at the cells, there is actually much more information that can be obtained from these tumor cells, because they really come from the tumor themselves. There's no exercise complicated bioinformatics exercise required in order to get all the noise sources removed. And there's actually even, let's say, different type of information available, such as the type of information contained in RNA. So what can we do in order to make sure that we have an easy and standardized workflow that can also isolate the cells? So this is where we think we have developed a new technology that can help doing that. And rather than explaining it to you in words, I want to try to show a movie which is an animation that was made in the context of this ERC grant. And if I manage to find my mouse, then there is no mouse. Okay, there it is. Then it should become visible to you guys. Oops, that's not what... Again, asking something very difficult from the...
Cancer is one of the leading causes of death, affecting millions of people across the globe. What makes cancer especially lethal is its spreading through the body, known as metastasis. Early detection of the highly aggressive circulating tumor cells responsible for the metastasis would allow us to treat cancer patients even before the cancer has spread. However, such circulating tumor cells are extremely hard to find. Typically, only one tumor cell can be found for every 10 billion red and white blood cells. We're literally searching for a needle in a haystack. IMEC has developed a technique that can rapidly analyze and distinguish different types of cells. The high-content, high-throughput cell imaging and sorting system is compact, low-cost, and easy to use. Through parallelization, systems that can inspect and sort up to 20 million cells per second are possible. The cell sorter is made up of 3 levels: the CMOS image sensor, the heaters, and the microfluidic channel, all fabricated in one production flow in IMEC's 200mm cleanroom facilities. The main elements of the cell sorter are the microfluidic channel, a simple light source, a CMOS imager, microheaters, and different in- and outlet channels. So how does our cell sorter work? When a blood sample is loaded into the system, the cells flow in via the main microfluidic channel. The sample contains a whole range of cells, several types of white blood cells, red blood cells, platelets, and maybe even some rare cells, such as circulating tumor cells. The microfluidic channel is about 30 microns wide, about the size of a single cell. Conditioned light illuminates the cells and results in fringe patterns that are recorded by the CMOS image sensor. These fringe patterns contain information on the 3D structure of the cell. The recorded holograms are then reconstructed into 2D images that are used to distinguish between the different types of cells based on cell size and nuclear morphology. These calculations, managed by an ASIC, need to be performed very quickly before the cells arrive at the microheaters further down the channel. The microheaters located at the crossing of outlets generate small and short-lived steam bubbles that gently but quickly push single cells to a particular outlet. While the red blood cells just go straight on, the white blood cells are brought to the lower outlet and the tumor cells are pushed in the upper outlet. The sorted cells can then be further analyzed downstream, for instance by extracting specific molecular information. After sorting, high-resolution 2D or even 3D images of each single cell can be consulted for further visual analysis. In our parallelized CMOS-based approach, up to 1,000 of these cell-sorting channels can be integrated in one compact system measuring a few square centimeters. By combining the most advanced techniques from various domains and by leveraging the enormous power of silicon nanotechnology, IMEC is revolutionizing the way life science research and diagnostics are performed.
So obviously, it's a movie, it's an animation, but I'm going to lead you through a few slides showing what the status actually is of this research. And we really believe this is an enormously promising technology that will come to you in a few years. So let's quickly explain what this lens-free microscopy is. So lens-free microscopy is a microscope without objectives or lenses. And therefore, it can be built in a super compact and easy way. So this microscope is less than 10 centimeters in a cube and has the possibility to have a similar performance as a conventional microscope, however, with a much larger field of view. So basically, every function in this microscope is replaced by a digital reconstruction algorithms. You can digitally zoom and get very similar information as with conventional microscopes, but with a much simpler system. So we use this lens-free microscope to look at a number of migration studies of metastatic cancer cell lines. And what we see is that indeed metastatic aggressive cancer cell lines move faster than other cell lines. But the most important point is here that we actually can use these lens-free techniques in order to very similar things than typically is done with conventional microscopes. And with the added advantage that the microscope is so small that it fits in the incubator. So you can actually constantly look at the cells. And there's no operators required. The focus and all the information that can be obtained from these images can be done by digital processing afterwards. So when we use the same lens-free microscope in order to look at cells which are flowing in microfluidic channels one by one, we actually can make those images in microfluidic channels. And so what you see on top is a hologram. So that is, let's say, a not digitally reconstructed version of the cell. But after digital reconstruction, you actually get a beautiful granulocyte flowing through this channel. And if we do that for PBMCs, we actually quickly discover that there's 3 populations of white blood cells present in our samples, and that is, of course, to be expected. These results also indicate that we actually are within 5% of what a Sysmex or other hematology analyzer can produce. But additionally, we have of each individual cell, we actually have the image available. So when we combine that with fluorescence information, we actually get even more information. We see subpopulations with even a better granularity. And so here we are looking at a 7-part classification, and the next step would, of course, be to start looking at how tumor cells would compare to those cell populations. So I said combining with fluorescence cytometry. Fluorescence cytometry can also be implemented on a chip by using photonic structures within the microfluidic chip. And here we show actually that we detect spikes from cells passing one by one in front of the detector, and which represent the fluorescent label. So that is how these pictures can be obtained. Can also easily be scaled up for multiple fluorescent colors, multiple fluorescent markers. And then if you combine all of that, so this lens-free cell imaging and this fluorescent cell detection into a cell sorter chip, where you can also sort the cells based on the detection signals that you get from the cells, you actually achieve what we want to achieve, namely a cell sorting chip which is much more powerful than what you have today in MAX and FAX technology, and where also the instrument can be implemented in a much more compact and cost-efficient way. So what does that have to do with circulating tumor cells? Well, we actually did the first experiments. And in the first experiments, we loaded MCF7 spiked PBMCs. And at the left side, you see what you normally see. So before sorting is actually stained in order to make visible the white blood cell. And if you look very hard, you actually will find one MCF7 cell, which is a model for breast cancer circulating tumor cells. Now, after we send this sample through our cell sorter, we collect the result, and we actually look at the sample again. And what you now see is a sample full of breast cancer or MCF7 cells, with here and there a white blood cell still present in the sample. So we have captured up to 94% of the cancer cells available in the original cell, and we have up to 70% purity of the cells, which is very interesting. Moreover, cell viability is well preserved, and so these samples can be sequenced immediately. So that is actually the technology that we have in development, and we are sure that there will be a continuation to this story, that we will try to translate this to the clinic, and I hope that you can keep watching us and in our continuation of this story. Thank you very much.
Thank you very much. Slightly mind-boggling. I remember going around your unit. It was like, goodness. Any questions from the floor? There's a question just there. I couldn't quite see you, but if you could take the microphone over. Fantastic.
Thank you very much. That sounded very exciting. My question, I have two questions actually. I can see where in clinical scenario where we could apply this, but would it be possible maybe to, wouldn't be that too late to wait for the cells to be in the bloodstream before picking cancer? This is my first question. My second question, can this be applied on cervical smear instead of using the traditional way of looking into abnormal cells? These are my two questions. Thank you.
Yeah, I'm not sure I understood the question
Sorry.
completely.
First question was like, isn't that to wait for these cancer cells to be in the bloodstream to look for them and pick them up? Is there any other way of maybe looking?
Okay.
That's my first question. My second, though clinically it's very important. I mean, I can see where we in clinical scenarios we can use this.
Yeah.
My second question, can this technology be developed around cervical screening? Looking for diskaryotic cells, for example.
Yeah, so I think the second question is a question for that I will refer to our host, because it's actually, I think, a medical, let's say partially a medical question for people that are in the field of cervical cancer. But the first question, of course, is it's one that is very easy to answer. So I think today what people in research often do is they use either MAX, which is magnetic bead-based capture of cells from blood, or FACS, which is flow cytometry. These are both tools which require many hands-on steps. Therefore, in different labs, you often get different results. They also don't produce the images, so they just capture the cells. What we believe is that by automating this, and also by capturing all these cell images, we have a way which can be much more easily standardized and compared across different labs. So it's automated and it's standardized, and that I think is something that is today lacking for most of the cell isolation or cell sorting tools around, and is very interesting for bringing something to the clinic. I think the second question, the application to the ovarian cancer,
cervical screening.
At this moment, if you look in the literature, there's more and more very recent papers coming out about circulating tumor cells in all types of cancer, also cervical cancer, but also in ovarian cancer patients. It seems that all stages together, stage 1 to 4, in roughly 60% of patients, there are circulating tumor cells. So even if the patient has a stage 1 ovarian cancer, there may be circulating tumor cells. So it might be well worth to look for them. You will not find them in all patients, obviously. And it's not because they're circulating tumor cells that there are metastases, but these cells have the potential to make metastases. So therefore, I think this is very new and very exciting research.
I got slightly lost at one point. I mean, using a CMOS image sensor, is that right? And I understood they could count, and separation, how does that work? Can you just very briefly?
Yeah.
So I thought that was more of a counting tool as a CMOS, but are you then a pressure or charge, or which way, how are you separating the cells?
Yeah, so the technology is very similar to an inkjet printhead, where you actually generate tiny droplets, steam bubbles, that push cells in certain direction. So steam bubbles are heat generated, but the heat never comes close to the cell. It's very far from the cell. So the cell only feels a mild push, kind of pressure, to go to one certain outlet.
It's a pressure thing. And then you recognize the cells using effectively some sort of pattern recognition, SVM, or some kind of automated feature extraction system, or how do you know what cells you've separated?
Yeah, so while they are flowing through the channel, we actually detect both the lens-free and the fluorescence signature. And those are in real time processed in order to steer the pressure and to send the cells to a certain outlet.
Okay, I think I'm a bit more with it. Sorry I'm catching up. Okay.
Yeah.
And I think we've got one more question. I think, is that right? Do you want to comment? Yes? A little comment. A pathological, I'm not sure if
Point
the
of view.
comment's pathological
I am
or
very
it's from a
interested to
pathologist.
hear the pathology.
No, only one brief comment.
Yeah.
I think that DNA, circulating DNA and tumoral cell are more informative than biopsy because circulating tumor cells and DNA are representative of the heterogeneity of the tumor, whereas biopsy is direct on the part of the tumor, and probably it doesn't represent the whole tumor. So in my opinion, this is the future. In
Are you putting yourself out of a job here, basically?
Yeah.
Perfect, okay.
I think additional to that, liquid biopsy, you can perform a liquid biopsy every month or every 2 months without any problem. So you can also look at the dynamics of the cancer much more easily than you can with a biopsy.
Okay, we're going to have to finish, Daniel. I'm really sorry because we have to stop because we are way over time. So I'm going to have to thank you very much indeed. Our final speaker of this session is Anne Coosemans, who is at KU Leuven working well, in fact, has been driving translational IOTA, really, I think, and amongst that is work on immunological markers. And she's going to present some new results from the immune side of things. I suspect we might have to have a long discussion with Dirk while we wait for the next computer to get linked up. I hope not. Maybe we can actually get this working straight away. I hold my breath.
Yeah, this. Is dat niet van Liesbeth deze? Denk het. Liesbeth.
Anne, how are you doing?
I'm fine, but I don't have much time, Tom?
No, you'll be fine. We'll just carry on. We'll just... We've lost time because of this fiddling around with computers, so we have to give people their fair share of time, unfortunately.
Okay.
Great.
Yeah, we're on. So good afternoon. I'm going to give you an overview of what Transiota now really is. You've heard a lot about it, bits and pieces. And then I'm going to show you a bit of the first results on Transiota, namely about the immune cell part. So the idea for Transiota actually came from, as you already heard, the role of the CA125 in the ADNEX model. So you can clearly see that, as it was already said today, for certain stages, for certain discrepancies, the addition of CA125 is good for the model. So we were wondering if it might be possible that we could add new biomarkers. So meaning that we could look for things in the blood, and then you come with the liquid biopsy that could give us information about the total tumor. And then I think to really know what type of biomarkers you should be looking for, you should really go into how cancer develops, how cancer progresses. And then you come, of course, to the famous paper of Hanaha, the hallmarks of cancer. For instance, the importance of immune system, the importance of genomic instability, as we already heard. I think you have to take these things into account when you determine what new biomarkers you can consider. So then we came up with the project of Transiota, and it's very easy for me to be the last one, but because then I don't have to introduce anymore the circulating tumor DNA, as Dieter Lambrechts already talked to you about, the circulating tumor cells, Liesbeth Lagae talked about them. We will always take care of the proteins. They were there early in the beginning. CA125 is a protein. They're easily accessible. It's very easy to process blood to determine proteins. You can determine proteins in huge amounts of samples at the same time. So yes, we will keep attention on the proteins, and then also the immune cells, but I will come back to that later on. So how is Transiota built? So we take a blood sample. Well, actually, we take 4 blood samples per patient. And from one blood sample, we isolate serum, from another one plasma, and from another one, we just take the whole blood. Serum is for the protein analysis, plasma is for the circulating tumor DNA, and whole blood we can use for the circulating tumor cells and the immune cells. This is the way we are doing it at the moment. So we actually aim for 1,000 patient samples, but they will not all be determined for immune cells, ctDNA, and everything. That would be very costly in a screening phase. So all of them will be screened for serum because, as I said, easy accessible, easy technique. And in 300 of them, we will do this new analysis, circulating tumor DNA, immune cells. To get this amount of patients, we will need the help of centers abroad, and we will also include 100 healthy controls. This is the situation in Leuven so far, so I think we already did our best. We have nearly achieved the inclusion for the immune cells because we only go there up to 200, and we are halfway for the healthy controls. This is a distribution of the samples as we have them today. So the majority is still benign, although, and yeah, of course, it is a cancer hospital, we have a lot of malignant samples. How are we going to do it, or how do we do it? So this part of the graph you already saw. So for protein analysis, we work together with Roche for commercially available proteins, but we will also make a more experimental setting into this immune parameters in serum, things that might be relevant for the future. The circulating tumor DNA is done in Dieter Lambrecht's lab. The circulating tumor cells, as you heard, in collaboration with IMEC, and the immune cells are my part, and I will come back to that. So, some results of the immune cells, but first of all, I'm going to take you through why the immune system is that important in the development of cancer. So, if you have normal tissue, then at a moment, a part of these cells will become neoplastic. We have discovered several factors, but we don't know them all. So several factors can play a role. Then the immune system comes into play. So a subset, different subsets of immune cells arise, and in best case scenario, they will eliminate these neoplastic cells. However, it's also possible that they don't succeed completely, and that the bad guys stay there, not active, but in kind of dormant phase, but they're not gone. And then, by several factors that we also don't know all at this moment, the tumor will reshape, and then actually the immune system gets total loss. So the immune control that was built is lost because the immune cells won't recognize a changing tumor anymore, and on top of that, immune suppression is drawn towards the tumor, meaning that certain types of immune cells will even aggravate tumor growth. To put it in another way, actually you have 2 types in the immune system. You have the very early innate immune system, and then you have the more precise adaptive immune system. So the innate immune system is there, is always there, is very rude, is without specification, but is always there immediately. The adaptive immune system, it takes about a week to get some adaptive immune response, but it's very precise, they know what to recognize, and they have memory. And in both categories, you have the good guys and the bad guys. So just remember especially this name, so it's part of the immune system, the MDSCs, part of the innate immune system, but actually an aggravator of tumor disease once MDSCs will develop in huge amounts. So again, a cartoon to show actually what is happening. So the tumor is there, the immune system comes into play, but then the bad guys, all the red ones, also come into play, and the tumor actually explodes. So for the immune part of the 178 patients, we have already analyzed 78 benign cysts with the distribution of the histologies like this, and 43 malignant patients, and 34 healthy controls. And then, thanks to a biostatistician, we have a lot of information. And then is, of course, the key to find where the exact information is that you need, because doing these things in blood for ovarian cancer and ovarian cysts, it has never been done before, so I cannot really compare with literature. But I can show you, I believe so, some nice graphs. So a fraction of the MDSC show a really nice trend upwards for invasiveness. So these are medians. These are the healthy controls, benigns, borderlines, invasive. So you can nicely appreciate that the more bad the ovarian cyst is, that the amount of MDSCs are going up. Remember, the MDSCs, part of the innate immune system, the bad guys, that is aggravating the disease. If you look more in detail, and we split out the invasive cancers, you can even nicely see that the stage 1, 2s are, within the invasive cancers, presenting with the highest values. So, which makes, of course, we are looking with new biomarkers, especially to discriminate these ovarian masses that we have difficulties with on ultrasound, being, for example, a borderline versus a stage 1. And you can see nicely how they seem or they tend to be discriminated based on these MDSCs. This is a slide I didn't need anymore. This one I needed. So you can look at it also in another way. This is an NMDS plot. So thanks to Peter, the bioinformatician. So it actually represent, the dots are the patients, and it gives actually a visual impression on how they behave one to another based on, again, the MDSCs. So it seems, but of course the results are preliminary and our sample size has to be increased, but there seem to be roughly 3 types of populations, the benign ones, the borderlines, and the invasive ones. These are only stage 1 and 2. Maybe there's also a role for NK cells. It's not so clear, I admit, as the MDSCs, but also here, invasive, borderlines, benign, they tend to be, they have a different median. Of course, another question is, can we differentiate in subpopulations within the benign group? I think for that, we don't have enough benign ones yet to tell this, but maybe already it seems, but of course only 5-4 samples, it's too little, but there seem to be trends. So, to conclude, the real setup for Trans-IOTA is that we still face diagnostic problems with ultrasound for stage 1 disease, the borderlines, to discriminate these diseases. And Trans-IOTA focuses on the detection of these new types of biomarkers. And certain types of immune cells really seem to behave differently between these different subtypes. So what do we plan in Leuven further? Yeah, to finish the inclusion. Then the startup in the centers abroad to go up to these 1,000 patients for serum collection. We will also search for links between circulating tumor DNA and immune cells. We will also search for links between the ultrasound parameters and these new biomarkers. And then I would like to thank, of course, a few people. So first of all, Dirk Timmerman for giving the opportunity for this type of research. Then my lab technician Anaïs, Chiara, and Thais for the huge amount of samples that they collect. Thais especially for this, her analysis of the immune cells, and Peter Busgaard, the bioinformatician, for all the results. Thank you.
Anne, thank you very much. Questions? Any comments? I mean, when you're talking about MDSC, I mean, what's happening? I mean, do you think that these cells are being stimulated? Are they being induced? Or is it an apoptosis or something? Are the cancers preventing them dying, or are they increasing their number?
Well, they are always there. So we have this immune suppression inside of us to not kill ourselves. So it's part of to avoid self-destruction. So we have these regulatory cells, we have these MDSCs, we have them, but at a normal threshold. And in cancer, they tend to increase, so proliferate and increase. And in the presence of cancer, they will stimulate even the cancer to grow.
But the point I'm making is that the cancer that's stimulating was the cancer, is the cancer inhibiting apoptosis and stopping them being killed naturally?
No, they also increase.
Yeah. And does that increase with tumor load, or is that just something that just happens? If you look at the tumor load, do the number of these cells increase, and so it's almost like a self-fulfilling prophecy that the cancer will spread because you increase the suppression of the body's immune system?
I don't know if I really understand.
Do they increase with tumor load?
Yeah.
Is that...
Well, we don't know much about MDSCs in ovarian cancer, so it is a subset of cells that has only been recently discovered, 2009, something like that, so the knowledge is not very broad about them, certainly not in ovarian cancer, but what we can observe is that they tend to increase with malignancy, and that within the stage of ovarian cancer, they also seem to be different. I think we don't have enough information yet today to tell why this is. Nobody knows, and I have not looked at it.
And is there a natural variation between individuals? So is there an inherent problem in some people?
There will certainly be but that's why you need, of course, several amounts of patients to limit out this variation. But yeah, you see these trends, and we can also for invasive cancers, because we follow them up over time, so after surgery, after chemo, when they recur, and you can couple the amount of MDSCs to the survival. So they really seem to play an important role in ovarian cancer as a driver mechanism.
Again, we could continue. I think we have to finish. Great talk. Thanks so much, Anne. Right, I'm well aware we're running terribly late, so thank you, Anne. Do you want to... Now, Dirk, how do you want to plan this? I would suggest that we obviously have coffee and take a 20-minute coffee break, and then we'll start. Eventually, at the end of the day, the drinks will start slightly late, but you still have an hour and 10 minutes of drinking. So I think that's enough for most people, even if you're English. So.
Okay, welcome back at the IOTA Congress. We are now having a coffee break. Perfect time to ask some small and very interesting questions to two speakers. I would like to welcome Lil Valentin from Sweden. She's a member of our steering committee. Lil, what's your first impression of this first IOTA Congress Day?
I'm overwhelmed. It's better than all my expectations. We have heard the latest news about IOTA 5. We have had a fantastic session on translational science with cell-free DNA, with tumor cells. It's really extremely exciting.
Okay.
I'm very happy that I'm here.
Thank you very much. You you will have a lecture in the afternoon. Can you give us a sneak preview on your own topic?
My own topic for this afternoon is ovarian cancer screening. I will give a short background, and then I will tell you the results of 2 randomized controlled trials on ovarian cancer screening and what they showed.
Okay.
Yeah, so that will be
very interesting.
Yeah.
Looking forward to.
Yeah.
So stay tuned. Thank you very much. I have another speaker next to me. This is Misty Blanchett Porter from the USA. Very welcome. I heard that this is your third time here at a Congress. What do you think about this third session? And can you tell why people in the future who are now watching from home should actually come to the Congress?
Yes, I'm very excited to be here. This is my third trip to Leuven. It's beautiful in terms of the scenery and the countryside here, but the magnitude of the science, the fellowship with others who are very interested in ultrasound and ovarian masses, and then cutting-edge research and the ability to look at the problem from many different aspects and integrate that knowledge so that we might approach our own patients at home with a different thought process and integrating that knowledge into how we might move forward in our own work.
Okay. Yes, thank you very much. Can you also give us a sneak preview of your topic, because you are our speaker also this afternoon?
Yes, I'm going to approach the American approach to the adnexal mass, and I can say where we would like to go or what we endeavor to do, because right now there are many different viewpoints about how we should approach the adnexal mass, but a lot of interest on the part of the American Institute of Ultrasound and Medicine and the American College of OBGYN into trying to get to a concerted way to triage patients with benign masses for observation and fertility conservation, but also getting masses of concern into the hands of an experienced sonographer for further triage, or directly to the GYN oncologist so they can get early expert treatment in the course of their ovarian cancers.
Mm-hmm. Okay, thank you. You will be here with us tomorrow. Are there other lectures you're looking forward to? I suppose you will see them all, but are there some speakers you say, okay, this is what people definitely should see and follow from home?
Yes, certainly the lectures by the IOTA Steering Committee I have also found extremely helpful. But tomorrow I'm very interested in the lectures on ovarian adnexal masses in pregnancy per se. Mm-hmm. Because right now I think...
Frédéric Amand, Professor Amand.
Yeah. Because right now in the United States, there's still a trend towards operating on masses in the second trimester, and there's a lot of information and data that say perhaps we should sit on our hands and choose expectant management to decrease intrapartum complications. So I'm very interested to see the work on pregnancy and adnexal masses tomorrow.
Okay. Thank you very much. Thank you both. I think coffee break is almost over, so we will go back to the Congress. Thank you very much.